So I'd like to welcome everyone today to our second April meeting. We have two great speakers lined up, Jimmy Moore and Denise Kirshner, and I hope we'll have a very lively discussion after their presentations. As I need to remind you, the meeting is being live streamed on YouTube. It's recorded, and those recordings will be made publicly available. As always, Reinhardt and I are the co-leads of the working group, and we have Jim Sluka and Bruce Shapiro helping us in all sorts of interesting ways. And of course, you've heard from them before. We're always available to do our best to try to deal with any issues that you want to have addressed with the working group. You've seen this slide many times. The Slack channel, the Twitter, the IMAG wiki page only work if you contribute to them. Please help us make them lively and useful. And we do encourage people to visit our YouTube channel and reference that. We have a great archive of talks now, and I hope those are useful to people and also can be distributed. So any ideas about how to make them more accessible and usable would be appreciated. Are there any announcements that people would like to make now that we have the short form announcements? Any people are free to announce conferences, meetings, funding opportunities, requests for help, anything. Okay, Jacob.
So I asked people from the working group, the reproducibility and credibility working group, and for the integration one, to look at their emails. There are several emails that are important that need some action from people. Please go and look at them and respond as quick as possible. Specifically, we have a representative from CISO, Catherine, who is willing to give us a talk. So please respond fondly to her. She's trying to help us.
Right. I sent her an email asking her if she would like to speak. Yes. Thank you very much for that introduction. I'll follow up again. If I don't hear back, I will follow up again.
Oh, you will. From Catherine, you will.
Any other announcements before we move on?
James.
TJ, go ahead.
Yes, thank you. Yeah, hi everyone. I'm TJ Seago. I work at the Biocomplexity Institute at IU with James. We are looking for opportunities to collaborate with clinicians and experimentalists on influenza infection and immune system modeling. Me and my collaborators with Erica Mochin, who's here today, from Carlo, Bard Ermintrout at Pitt, and then James as well. We've developed a multi-scale, multicellular spatial model of influenza infection and immune response that was derived from an ODE model that Bard and Erica formulated and then calibrated to murine data of influenza infection. It has lots of relevant biological objects and processes associated with the immune response. So I'm interested in discussing potential opportunities to couple computational modeling and simulation using this model with experimental work. So if anyone would be interested in having such a conversation, you can contact me at tjseago@iu.edu. I'll put that in the chat as well. And that's it. Thank you very much, James.
Okay, thank you. Any other announcements, comments? Okay, it's the comments and announcements section of the talk is new, so I guess it'll take us a few weeks to get used to having that opportunity. Thank you. Okay, another reminder that one of the outputs on which we are judged are collaborations. And so if this working group has been useful to you in any way, in particular, writing a paper with somebody, preparing a grant proposal with somebody, teaching a modeling resource, just extensive discussions with people you might not have met otherwise, please do let us know so that we can put that in the record and explain and justify our existence. That would be very helpful. Upcoming meetings. Next week, we have Winston Garira from South Africa and Vivek Shenoy from University of Pennsylvania. April 22nd, we have Deborah Peters, who's going to be talking about very multi-scale modeling of animal epidemics. Very interesting and important work. And Then April 29th, we have Penelope Morel speaking, I assume on immunology. And we have an open slot. And so if somebody from one of the subgroups would like to present in that open slot, we would love to have a presentation from one of the subgroups. If not, we are open to suggestions. And we have a variety of talks coming up. We're going to have a talk from one of the developers of the original digital twin concept coming up a little later in the summer. As always, we welcome your suggestions for additional speakers. Be very grateful for that. As usual, during the meeting proper, we would appreciate short questions and ones from people who don't normally ask the questions, and more general questions. We're going to have the nice half hour after the meeting, which I hope many of you can stay for. We'll do it open format where Denise and James Moore will stay in the main meeting, and they'll be able to talk to each other and to anybody who has questions. That worked really well last week. I hope it's productive this week as well. And if people look like they may be running overtime a little bit, I'll give a 5-minute break into the speakers and give a 5-minute warning so we stay on time. I know it's very hard to keep presentations into a 20-minute slot. They're really teasers and opportunities. You're always welcome to come back. Denise and James, you're welcome to come back and give another talk with more detail. We most welcome. Okay, so without more ado, we're going to move on to our first mini seminar. James Moore, I guess you said Jimmy Moore is your preferred salutation?
Yeah, Jimmy, please, it's fine.
And on biotransport mechanisms for adaptive immunity. And people know that we're informal here, so we don't do big introductions. So I will just say, take it away, please.
Right, thank you very much. It's a great honor to be here, and I assume I'm free to start sharing the slides. So I'm really pleased to be part of the program this evening. I've tried to dial in a few times to these things, and it's just not at a great time of a day here in London. But I'm really appreciative to the group and for inviting me and for organizing these things. Second of all, I'm honored to be the opening act for Denise Kirschner, and I think her research has been a great inspiration to a lot of what we've been looking at in lymph nodes, and look forward to hearing what she has to say this evening. So the first slide gives you a bit of a laundry list on the left of some of the research going on in my lab. The dark characters show the ones that are related to the lymphatic system, and really, yeah, it's about 2/3 of what goes on in the lab. The stuff in gray is the other things. We do modeling, which you'll hear about today. We do experiments, which you'll hear a little bit about today. But I'm always pushing my PhD students, my postdocs, to think about what the results are telling them, and try and come up with device ideas or ideas that can impact patient care, and at the end of the day, that's what justifies our existence, and so we're always looking for ideas to come out of that, and as part of that, I actually run a medical device entrepreneurship program here. It's a master's program, which is a lot of fun, but I'm not going to talk about that at all. So really, my aim this evening is to kind of give a conversation starter, a bit heavy on questions that we haven't answered yet, and a bit of results of the things that we have answered that we think we know. And so I'll start out with a bit of an introduction to the lymphatic system. I find we know that it's not taught as well as it should be in medical schools, and that's true around the world. So a bit of an introduction here. Its main job is fluid balance, going back from the periphery into the blood circulation. So you have a net leakage of fluid out at the capillary level, and that fluid is an important part of nutrition and fluid balance and tissue health. And so that fluid's got to be gathered up and eventually returned to the blood system, or you end up with swollen tissues. And that's the condition lymphedema. Now, all of the fluid that comes into here, the initial lymphatics are very porous, and the fluid that comes in may have dirt and debris, may have cancer cells. So that fluid needs to be filtered and cleaned and before it can be sent out back around the whole body in the blood system. That's the job of lymph nodes. You have about 500 or 600 of them in your body. And there are specialized blood vessels inside lymph nodes, which we should not ignore. They're very important. And it looks like metastatic cells that come into the lymphatic system will preferentially go into the lymph node blood vessels to get to the blood system. They less commonly continue on in the lymphatic system, which is an interesting phenomenon that was discovered a couple of years ago by Tim Padera and some other researchers. So lymph nodes, hugely important. Now, in terms of fluid mechanics, so I started out as a mechanical engineer, and so I like fluid mechanics. So blood flow, you get about 5 liters a minute cardiac output. Blood pressure, again, you probably know these numbers. Aortic diameter is 2 to 3 centimeters. The whole lymphatic system only pumps about 5 liters per day. So very low flow system. The biggest lymphatic vessels, about 2-3 millimeters in diameter. And the pressures, that's not a misprint. The pressures vary from very far subatmospheric up to basically venous pressure, and which is a very interesting phenomenon of lymphatic flow to look at. So you think about the lymphatics that drain the pleural side of the diaphragm. You got to keep your lungs inflated, so that's subatmospheric pressure already, and then you got to drain fluid out of that, so it's even lower. So so those are the basic numbers of the system. Now, what I'd like to talk about today is a sort of a teaser and a survey of what we've done in terms of adaptive immunity. So whether it's a vaccine or an infection that comes in through the skin, the job of the lymphatic system is to, or the immune system in general, is to recognize it and then transport that information into the lymph node. So the key to the lymph node is that there's a high enough density of the proper immune cell types to develop adaptive immunity. So antigen recognition will happen in the periphery, but really, you know, the process of passing that information on to other immune cells, it requires the cell densities that you find in lymph nodes. So you look at the intradermal space, subcutaneous, intramuscular space. You have lymphatic vessels, initial lymphatics, to varying degrees in these tissues. And then what I'm going to spend a bit of time talking about first is these collecting lymphatic vessels that do the job of the pumping, of delivering that information into the lymph node. And then, of course, once that information is processed, it can go into the specialized high endothelial venules. These are particularly good at passing immune cells back and forth. And so that some of that information can go into the HEV, some of it can go into the efferent lymph, but it all ends up back in the systemic blood, and then that information gets passed around the body. So the next time you encounter the infection, then you know what to do about it. So let's talk a little bit about timescales. So we know that when antigens are delivered into, say, the subcutaneous space or even intramuscular space, they start showing up in the lymph nodes within minutes. And these lymph nodes might be several centimeters away. So, and this has been recognized in multiple animals, multiple lymph nodes, multiple injection locations. And one thing I find as I talk to other types of scientists about this is they keep asking me, what about cell motility? So don't antigen presenting cells get the information and then sort of crawl their way to the nearest lymph node? Well, not really. Antigen presenting cells do actively crawl through the interstitium. They follow chemokine gradients to find their way into a lymphatic vessel. They crawl along lymphatic vessel walls in the initial lymphatics, and that appears to be chemokine triggered as well. But the velocity they can reach is about 5 nanometers per minute. And if you do the unit conversion in your head, you can see that it would take about 4 years to traverse a distance of 1 centimeter at that velocity. So this is not a realistic option. Let's say if you get an infection in your toe, the nearest lymph node is in your knee. So the cell crawling there is just not going to work. So the body has done for the immune system what it has done for other systems. When you need to get something there fast, you learn to do fluid mechanics. And I'll just add, even if the antigens are coming in suspension, the diffusion it would be similarly limited to a very long time scale. So this is how it happens. This is a rat mesenteric lymphatic vessel. The mesentery is chock full of lymphatics. You can stretch the mesentery of a rat over a microscope stage and you quickly find lymphatics. This one is about 100 microns in diameter. It's about the diameter of a human hair. These are really the workhorse vessels of the lymphatic system. They do pretty much all of the pumping. You can see some squeezing here, and that's true. There are check valves just upstream and downstream of this image that keep the flow moving in the right direction. So within that lymphatic vessel, you will see cells floating through there. So those are either lymphocytes or dendritic cells, so the white blood cells in your body. And they are achieving a velocity about 1 centimeter per second. And if you're keeping score, that's 8 orders of magnitude faster than they would be able to crawl. So, so this is how we get information to the lymph node from the periphery. The flow rate is about 200 microliters per hour. And I remember when my student, Brandon Dixon, who did these measurements, when he brought that to me, I said, wait a minute, did you just say we measured flow in microliters per hour? And he said, yes. And I was really pleased by that. It's a great unit of flow. But on the other hand, the peak wall shear stress ends up being about the same in your lymphatic vessels of these size as it is in the aorta. So 5 liters a minute going through your aorta will produce about 5 dynes per square centimeter of shear stress. So interesting fluid mechanics there for sure. Now, once that information gets to the lymph node, it comes in these afferent vessels. There are typically multiple afferent vessels feeding an individual lymph node. There is a subcapsular sinus around the periphery where the fluid comes in. There are B cell follicles. Here there's a T cell cortex. There's a medullary sinus, which is thought to be sort of the collection point for dumping fluid into the efferent vessels. And if you look at sort of the flow pattern of how flow might come in, so it comes in from the afferent vessels, it can go into the subcapsular sinus. It actually might go around the periphery of the lymph node directly into the medullary sinus. Those connections have been recognized over and over again. Or it can go through the sort of porous medium part of the lymph node, the B cell follicles, T cell cortex, and then there are blood vessels actually scattered through all of this, where important fluid chain exchange happens as well. Now that subcapsular sinus is sort of an open region. It's like a parallel plate flow situation if you're more of a fluid mechanics person like me. The height of the subcapsular sinus is on the neighborhood of sort of 10 to 20 microns. And so if there's an antigen presenting cell that comes in, it'll sort of sit in the subcapsular sinus and perhaps pass that information on to the cells that line the basement of the subcapsular sinus here, or pass that or directly crawl in. And when they do, they follow chemokine gradients within the lymph node as well. So chemokine gradients are important not only in the periphery but also in lymph nodes. Now, so we wanted to know a bit more about the flow in these lymph nodes, how the lymph is distributed when it comes in. It's very difficult to measure. The Peclet number, which is sort of the ratio of diffusive to convective effects, is on the order of unity in these vessels. So if you put a fluorescent dye into a lymph node, it'll diffuse about as fast as the fluid would move it. So it's actually hard to figure out what the flow velocities are. So we constructed a computational model, and using our the best measurements we could find, some measurements we did ourselves, and of course the need to keep the pressure differences realistic, because keep in mind all of the flow through the lymph node is being driven by the afferent vessels, which can only generate a couple of centimeters of water of pressure. So it turns out most of the flow actually goes around the periphery. So this is remember the subcapsular sinus is this open region. It's very low, has a very low resistance to flow compared to the sort of meaty, porous media region of the lymph node. The medullary sinus is also relatively low resistance, somewhere in between the subcapsular sinus hydraulic conductivity and that of the paracortex. So that's the path of least resistance. That's where most of the flow goes under baseline healthy conditions when there is no response to antigen going on. Now this flow will deliver antigens, so antigens can be directly suspended into the lymph and come in, and some of that will certainly go into the T cell cortex here, into the B cell follicle regions, and that can occur along some interesting structures, these collagen conduits that exist in the lymph nodes, and there's a size selective filtering. It's an absolutely fascinating little chemical engineering factory inside these lymph nodes, and hopefully Denise will tell us a lot more about that. Also, these flow patterns are important in determining the chemokine gradients in lymph nodes. So if you try and predict what chemokine concentrations are in the lymph node with just diffusion, you will go wrong. You will go horribly wrong. You need to incorporate the fluid flow in order to reconstruct what these chemokine gradients are inside the lymph node. And again, that's another interesting chemical engineering problem. So one thing that we did was we subjected some lymph nodes to, these are actually vaccine adjuvants, LPS and poly-IC. And we looked at the resistance, the flow resistance. So this is the pressure drop across the lymph node divided by the flow rate through the lymph node. And remember, these flow rates are maybe 100 microliters per hour. The pressure differences are a couple of centimeters of water. These are exquisite little measurements to do. And so what we find is about an order of magnitude increase in flow resistance in these lymph nodes within 24 hours, and within 72 hours, it goes back to baseline. And so your time scale for lymph node swelling, which is something you're probably familiar with when you get sick, your lymph nodes swell. Lymph nodes will swell typically 24 to 48 hours after the infection. So this is really the resistance to flow goes up a lot faster than the lymph node swells, and then goes back down probably before the lymph node is finished doing all of its swelling. So this got us puzzled. If the thing's not changed much in size yet, what about this is causing a change in flow resistance? So then we went back to the computational model and we started varying things, as you do with the computational model. So the first thing they did was vary the hydraulic conductivity of the T cell cortex.
Remind you, 5 minutes.
Okay, thanks. Now, but remember, only 10% of the flow is coming into the middle here under baseline conditions. And with 90% of the flow going around this low resistance pathway here, you could change that hydraulic conductivity all you want. It does nothing to the overall flow resistance of the lymph node. We also changed the height of that subcapsular sinus. We doubled it, we halved it, we did all kinds of things to it. And we could only produce about that much change in the overall flow resistance of the lymph node. The next thing we did was we changed the hydraulic conductivity of the medullary sinus. So that's this sort of collection point down at the bottom here. It is part of the low resistance pathway. So not surprisingly, I guess, if you change the hydraulic conductivity of the medullary sinus by an order of magnitude, which would not be hard to do in a porous medium, then you end up being able to explain all of the change in the overall flow resistance of the lymph node. That's the only part of the lymph node that we changed that produced anything close to the flow resistance change that we measured in the experiments. So that is what we think is causing the change. We suspect there is a specialized kind of cell in the medullary sinus that when it senses an antigen to respond to, that it shuts down that low resistance pathway, sends more of the flow into the middle. So it about triples the flow into the T cell cortex when it shuts down that low resistance pathway, which, I mean, all this makes sense, but I guess we had to sit around and do some modeling and some experiments to figure it out. So now these are, as I mentioned, this poly-IC and LPS, they're sometimes used as vaccine adjuvants in animal experiments at least. So I thought I'd end the talk with talking a bit about what we think about vaccine adjuvants. So I'll give a bit of history. Like many good medical stories, the history begins in London here. So there was Alexander Glennie was doing some experiments in the south part of London. on vaccines, and he noticed that when they showed signs of inflammation around the injection site, that the vaccine worked better. So he started sort of inducing inflammation around the injection site by adding aluminum salt or alum to the vaccine. And he found it worked. And as things went in the 1920s, if something worked, then you just walked over to the human hospital and you started injecting people, and away you go. We probably all have had alum injected into us as part of vaccines that we've had. It's been used millions of times, literally, and it continues to be the most common vaccine adjuvant. Funny thing is, nobody really knows how it works, whether it works in the periphery, whether it works in the lymph node. Glennie himself proposed something called the depot theory, which is that it irritates things around the injection site to the point where more immune cells come there. There's actually not much evidence for that. So since then, there's very little support for the depot theory. It is still the dogma in the field. And I've had immunologists get angry with me for suggesting that it may not be the case. There are two additional types of adjuvants that have been developed: oil and water emulsions, TLR activators. TLR activators are very effective, but they're toxic. The ones that work well in mice don't necessarily work well in humans. Nobody really knows how they work either. So if you think about the job of a vaccine designer, how do you pick one? And the answer is nobody knows. They just kind of try one, and if it seems to work, then they go with it. I'm not going to show the results today, but I'll tell you, our preliminary results show that all three types of adjuvants actually shut down that lymphatic pumping. That's that squeezing of those collecting vessels that delivers the information from the periphery into the lymph node. So if you're trying to get information into the lymph node quickly, using an adjuvant's not the way to go. So let's go back to our picture here then. So let's think about what's the job here. The job is to deliver the information from the syringe, get it into the lymph node where there's a high enough density of immune cells to do something about it. So we'd like to know, sort of starting with the lymph node, I think is the right way to go here. So what temporal profile of antigen delivery does the lymph node need in order to do its job? And so step one, peripheral uptake. All right, well, we're injecting them intramuscular where there are not many lymphatics. Step two is to get them into the prenodal lymphatics, and we know that adjuvants downregulate lymphatic pumping. So what's that do for us? Well, if we're trying to slow things down, then our first two steps are doing the right thing, but don't know if that's the right thing to do. And then within the lymph node, our results show that it increases the flow into the T cell zone to add these adjuvants in. So what does that all do? We don't know. We'd like to know more about this system so we can provide for the vaccine manufacturers a platform for designing vaccine adjuvants and deciding which one should go with their particular antigen. So just wrapping up the lymphatics, it's not a commonly studied system, which makes it the land of opportunity for me and other people that are crazy enough to take it on. The lymphatic pumping, we've done a lot of work on this over the years, and it's a fascinating fluid mechanics problem. And that's the only way you get information into your lymph nodes quickly enough to do anything about it. And again, I think we need, for vaccines, we need to start with the lymph node. What does it need? What time course does it need in terms of antigen delivery, and how can we design adjuvants that do that? So with that, I'll close with acknowledgements to the people that really did the work, and great collaborators and great funding sources. So thank you. And I look forward to hearing what Denise has to say.
Okay, thank you very much. I will, if it's okay, I think we'll hold the questions to the end.